Sanger sequencing is a method of DNA sequencing based on the selective incorporation of chain-terminating dideoxynucleotides by DNA polymerase during in vitro DNA replication. Developed by Frederick Sanger and colleagues in 1977, it was the most widely used sequencing method for approximately 25 years. More recently, Sanger sequencing has been supplanted by next-gen sequencing methods, especially for large-scale, automated genome analyses. However, the Sanger method remains in wide use, primarily for smaller-scale projects and for obtaining especially long contiguous DNA sequence reads. Method: the classical chain termination method requires a single-stranded DNA template, a DNA primer, a DNA polymerase, normal deoxynucleoside triphosphates, and modified dideoxynucleotide triphosphates, the latter of which terminate DNA strand elongation. These chain-terminating nucleotides lack a 3-OH group required for the formation of a phosphodiester bond between two nucleotides, causing DNA polymerase to cease extension of DNA when a modified ddNTP is incorporated. The ddNTPs may be radioactively or fluorescently labeled for detection in automated sequencing machines. The DNA sample is divided into 4 separate sequencing reactions, containing all 4 of the standard deoxynucleotides and the DNA polymerase. To each reaction is added only one of the 4 dideoxynucleotides, while 3 other nucleotides are ordinary ones. Putting it in a more sensible order, 4 separate reactions are needed in this process to test all 4 ddNTPs. Following rounds of template DNA extension from the bound primer, the resulting DNA fragments are heat denatured and separated by size using gel electrophoresis. In the original publication of 1977, the formation of base-paired loops of ssDNA was a cause of serious difficulty in resolving bands at some locations. This is frequently performed using a denaturing polyacrylamide urea gel with each of the 4 reactions run in one of 4 individual lanes. The DNA bands may then be visualized by autoradiography or UV light, and the DNA sequence can be directly read off the X-ray film or gel image. In the image on the right, X-ray film was exposed to the gel, and the dark bands correspond to DNA fragments of different lengths. A dark band in a lane indicates a DNA fragment that is the result of chain termination after incorporation of a dideoxynucleotide. The relative positions of the different bands among the 4 lanes, from bottom to top, are then used to read the DNA sequence. Technical variations of chain termination sequencing include tagging with nucleotides containing radioactive phosphorus for radiolabeling, or using a primer labeled at the 5' end with a fluorescent dye. Dye primer sequencing facilitates reading in an optical system for faster and more economical analysis and automation. The later development by Leroy Hood and co-workers of fluorescently labeled ddNTPs and primers set the stage for automated, high-throughput DNA sequencing. Chain termination methods have greatly simplified DNA sequencing. For example, chain termination-based kits are commercially available that contain the reagents needed for sequencing, pre-aliquoted and ready to use. Limitations include non-specific binding of the primer to the DNA, affecting accurate readout of the DNA sequence, and DNA secondary structures affecting the fidelity of the sequence. Dye-terminator sequencing. Dye-terminator sequencing utilizes labeling of the chain terminator ddNTPs, which permits sequencing in a single reaction, rather than 4 reactions as in the labeled primer method. In dye-terminator sequencing, each of the 4 dideoxynucleotide chain terminators is labeled with fluorescent dyes, each of which emit light at different wavelengths. Owing to its greater expediency and speed, dye-terminator sequencing is now the mainstay in automated sequencing. Its limitations include dye effects due to differences in the incorporation of the dye-labeled chain terminators into the DNA fragment, resulting in unequal peak heights and shapes in the electronic DNA sequence trace chromatogram after capillary electrophoresis. This problem has been addressed with the use of modified DNA polymerase enzyme systems and dyes that minimize incorporation variability, as well as methods for eliminating dye blobs. The dye-terminator sequencing method, Along with automated high-throughput DNA sequence, the analyses is now being used for the vast majority of sequencing projects. Automation and sample preparation. Automated DNA sequencing instruments can sequence up to 384 DNA samples in a single batch and up to 24 runs a day. DNA sequencers carry out capillary electrophoresis for size separation, detection and recording of dye fluorescence, and data output as fluorescent peak trace chromatograms. Sequencing reactions by thermocycling, cleanup and resuspension in a buffer solution before loading onto the sequencer are performed separately. A number of commercial and non-commercial software packages can trim low-quality DNA traces automatically. These programs score the quality of each peak and remove low-quality base peaks. The accuracy of such algorithms is below visual examination by a human operator, but sufficient for automated processing of large sequence data sets. Challenges. Common challenges of DNA sequencing with the Sanger method include poor quality in the first 15 to 40 bases of the sequence due to primer binding and deteriorating quality of sequencing traces after 700 to 900 bases. Base calling software such as FID typically provides an estimate of quality to aid in trimming of low-quality regions of sequences. In cases where DNA fragments are cloned before sequencing, the resulting sequence may contain parts of the cloning vector. In contrast, PCR-based cloning and next-generation sequencing technologies based on pyrosequencing often avoid using cloning vectors. Recently, one-step Sanger sequencing methods such as Amples-Can-Sec-Sharp have been developed that allow rapid sequencing of target genes without cloning or prior amplification. Current methods can directly sequence only relatively short DNA fragments in a single reaction. The main obstacle to sequencing DNA fragments above this size limit is insufficient power of separation for resolving large DNA fragments that differ in length by only one nucleotide. Microfluidic Sanger sequencing. Microfluidic Sanger sequencing is a lab-on-a-chip application for DNA sequencing, in which the Sanger sequencing steps are integrated on a wafer-scale chip using nanoliter-scale sample volumes. This technology generates long and accurate sequence reads, while obviating many of the significant shortcomings of the conventional Sanger method by integrating and automating the Sanger sequencing steps. In its modern inception, high-throughput genome sequencing involves fragmenting the genome into small single-stranded pieces, followed by amplification of the fragments by polymerase chain reaction. Adopting the Sanger method, each DNA fragment is irreversibly terminated with the incorporation of a fluorescently labeled dideoxy chain-terminating nucleotide, thereby producing a DNA ladder of fragments that each differ in length by one base and bear a base-specific fluorescent label at the terminal base. Amplified base ladders are then separated by capillary array electrophoresis with automated, in situ finish linear detection of the fluorescently labeled ssDNA fragments, which provides an ordered sequence of the fragments. These sequence reads are then computer-assembled into overlapping or contiguous sequences which resemble the full genomic sequence once fully assembled. Sanger methods achieve read lengths of approximately 800 bp. The longer read lengths in Sanger methods display significant advantages over other sequencing methods, especially in terms of sequencing repetitive regions of the genome. A challenge of short-read sequence data is particularly an issue in sequencing new genomes and in sequencing highly rearranged genome segments, typically those seen of cancer genomes or in regions of chromosomes that exhibit structural variation. Applications of microfluidic sequencing technologies. Other useful applications of DNA sequencing include single nucleotide polymorphism detection, single-strand conformation polymorphism heteroduplex analysis, and short tandem repeat analysis. Resolving DNA fragments according to differences in size and/or conformation is the most critical step in studying these features of the genome. Device design. The sequencing chip has a four-layer construction, consisting of three 100 mm diameter glass wafers and a polydimethylsiloxane membrane. Reaction chambers and capillary electrophoresis channels are etched between the top two glass wafers, which are thermally bonded. Three-dimensional channel interconnections and microvalves are formed by the PDMS and bottom manifold glass wafer. The device consists of 3 functional units, each corresponding to the Sanger sequencing steps. The thermal cycling unit is a 215 nL reaction chamber with integrated resistive temperature detector, microvalves, and a surface heater. Movement of reagent between the top all-glass layer and the lower glass PDMS layer occurs through 500 1/4 m diameter via holes. After thermal cycling, the reaction mixture undergoes purification in the capture purification chamber, and then is injected into the capillary electrophoresis chamber. The CE unit consists of a 30 cm capillary which is folded into a compact switchback pattern via 65 1/4 m wide turns. Sequencing chemistry, thermal cycling, in the TC reaction chamber, Terminator sequencing reagent, template DNA, and primers are loaded into the TC chamber and thermal cycled for 35 cycles. Purification, the charged reaction mixture is conducted through a capture purification chamber at 30 degrees Celsius via a 33 volts per centimeter electric field applied between capture outlet and inlet ports. The capture gel through which the sample is driven, consists of 40 1/4 m of oligonucleotide covalently bound to a polyacrylamide matrix. Extension fragments are immobilized by the gel matrix, and excess primer, template, free nucleotides, and salts are eluted through the capture waste port. The capture gel is heated to 67-75 degrees Celsius to release extension fragments. Capillary electrophoresis, extension fragments are injected into the CE chamber where they are electrophorezed through a 125-167 volt per centimeter field. Platforms, the Apollo 100 platform integrates the first 2 Sanger sequencing steps in a fully automated system. The manufacturer claims that samples are ready for capillary electrophoresis within 3 hours of the sample and reagents being loaded into the system. The Apollo 100 platform requires sub-microliter volumes of reagents. Comparisons to other sequencing techniques, the ultimate goal of high-throughput sequencing is to develop systems that are low-cost, and extremely efficient at obtaining extended read lengths. Longer read lengths of each single electrophoretic separation, substantially reduces the cost associated with de novo DNA sequencing and the number of templates needed to sequence DNA contiguous at a given redundancy. Microfluidics may allow for faster, cheaper and easier sequence assembly. References. Further reading, http nanocancer.gov Nanotech News 2006-05-30 ASP, http nanocancer.gov Monthly Feature 2005 OGASP, external links, MBI says new tool that automates Sanger sample prep cuts reagent and labor costs.